Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article entitled MAP plus maintenance pegylated interferon 2B versus MAP alone in patients with resectable high-grade osteosarcoma and good response to preoperative MAP. First results of the Euramos 1 good response randomized controlled trial by Stefan S. Bielak et al. My name is Carola Arndt, and I am a pediatric hematologist-oncologist and professor of pediatrics at Mayo Clinic in Rochester, Minnesota, USA. My oncology specialty is pediatric oncology. Bielak and colleagues should be congratulated on a successful effort in launching and completing a multinational, multi-institutional, collaborative clinical trial involving 326 centers from 17 countries. This article focuses on the first results of the Euramos 1 good response randomized controlled trial. Although osteosarcoma is rare, with an incidence of only 2 to 3 persons per million per year, it is still the most common malignant bone tumor in children and adolescents. Standard chemotherapy remains methotrexate, doxorubicin, and cisplatin, referred to as MAP throughout this podcast. One of the most important prognostic factors is histologic response to preoperative chemotherapy. Patients with less than 10% viable tumor in the resected specimen, so-called good responders, have a 5-year survival of approximately 75 to 80%. With a 5-year survival this good, it is not justified to study addition of toxic chemotherapy agents with more short and long-term side effects for the subgroup of good responders. The investigators chose to study the use of interferon after completion of maintenance chemotherapy in good responders with the rationale provided in the background section of the manuscript. A total of 2,260 patients were registered on the study, of which 1,041 were confirmed good responders. Only 716 of those were actually randomized, 69%. The main reason for non-randomization being patient choice. Of the 357 patients randomized to start interferon, only 76% actually ever started it, and of those, only 47% completed it at the time of this report. Refusal was the main reason for never starting interferon, and toxicity was the main reason for stopping early. Although the results showed the hazard ratio was 0.83 from adjusted Cox model in favor of interferon, the authors concluded that MAP interferon was not statistically different to MAP and that current therapy for good responders should not change. The major shortcoming of the study is the high rate of noncompliance with allocation of treatment to the interferon arm, as well as the high rate of treatment abandonment in those who did not start interferon. This may have resulted in underpowering of the ability to detect a difference, but the clinical reality remains that if one cannot convince patients to do the therapy they're assigned to, there is little point in pursuing that approach. Curiously, patients who were assigned to interferon but did not start it had an inferior outcome to those who were not assigned to it. It is unknown if there were different rates of MAP toxicity between those patients who did or did not start interferon after being assigned to it, or in those who refused randomization. There are many other subsequent analyses that I anticipate will be forthcoming from this study. The formidable logistics of executing such a study are well described by Dr. Nessa Marina in the journal Cancer Treatment and Research, Volume 152, 2009, from which I will utilize detailed examples. Given the rarity of osteosarcoma and the plateau in event-free survival of 60 to 70%, large numbers of patients are required to detect further improvement in outcome. 4 multinational groups agreed to cooperate and collaborate in this effort. These groups were the Children's Oncology Group, the Cooperative Osteosarcoma Study Group, the European Osteosarcoma Intergroup, and the Scandinavian Sarcoma Group. While the concept and protocol document were developed within 18 months, approximately 4 years elapsed before activation of the final protocol in all countries. Initially, United States regulatory bodies required each non-North American participating institution to obtain a federal-wide assurance number. After recognition of the overwhelming burden of this condition, it was decided that the overall European medical sponsor, the Medical Research Council in the United Kingdom, be allowed to renew their federal-wide assurance. Fortunately, the safety desk in Münster, Germany, already had a federal-wide assurance number due to Children's Oncology Group participation in one part of the Euro-Ewing study, and was able to assume responsibility of serving as the safety desk of the Euramos study as well. The Medical Research Council Clinical Trials Unit functioned as the data coordination center, as it might have been problematic to transfer European data to centers outside of Europe, and common data collection points were agreed upon. The requirement for an independent data monitoring committee was also problematic because international guidelines preclude membership in the data monitoring committee of any investigator participating in the study, and finding qualified non-participants with no conflicts was challenging. The European groups had not been previously required to conduct quality control through on-site audits, and procedures were implemented to make this happen in Europe. Standardization of chemotherapy administration guidelines was a hurdle also, in particular administration of doxorubicin to decrease risk of cardiotoxicity. Since dexrazoxane is not licensed in Europe, it was mutually agreed upon to administer doxorubicin by continuous infusion. Pegylated interferon is not licensed for the indication in this study, and investigators cross-filed on the IND application of sharing plow and developed procedures to distribute drug to the various countries. Sharing plow kindly provided drug at no cost. Finally, all of this effort requires funding, which is also time-consuming and difficult to obtain. A full listing of the funding sources is provided in the appendix and is summarized by the following author's comment, quote, "Funding for this academic trial was provided by governmental and charitable bodies in each participating country, with additional funding for pan-European trial coordination," end quote. This study involved working together across oceans, regulatory bodies in various countries, different cultures in 4 cooperative groups, different standard chemotherapy administration guidelines, and putting aside personal biases and styles to work together and come up with a document acceptable to all in order to conduct an international trial in a rare disease for the benefit of children with osteosarcoma. International cooperation is indeed feasible, and this study is an example for future efforts in rare diseases. Other examples in pediatric malignancies include ANHL 1131 for B-cell non-Hodgkin's lymphoma and parts of Euro-Ewing 99. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.